Hello and welcome to the mind map about cell membranes and the transport through cell membranes. All right, take a snapshot now if you want to, otherwise we're zooming in. All right, we come up here to the composition. Cell membranes are made of phospholipids, and phospholipids have a phosphate head and fatty acid tails. The cell membrane is a phospholipid bilayer, which means it has two layers of phospholipid. Here is a diagram of the cell membrane. The cell membrane adheres to the fluid mosaic model. It's a fluid because all of the phospholipids move independently of each other and are fluid in that they are moving. Cholesterol provides stability to the fluidity so that the membrane doesn't fall apart. The mosaic refers to there are many different components within the cell membrane, being lipids and proteins and glycoproteins and lipoproteins and cholesterol. All right, inside the membrane, we've got alpha helix proteins, and they are what we call transmembrane proteins, in that they link the outside to the inside. The outside of the cell is called extracellular fluid. The inside of the cell is called intracellular fluid. There are different types of alpha helix proteins, being ion channels that allow the passage of ions through the membrane from one side to the other. There are ion pumps. We also have other things called glycoproteins, which are proteins that have carbohydrate tails sticking out of them. There are also lipoproteins that have lipid tails attached to the proteins that are embedded in the membranes. And glycoproteins and lipoproteins function as either receptors or MHC molecules, but they are used for cell communication. All right, there is a picture of the cell membrane and the phospholipid bilayer there. It's showing that the heads are hydrophilic, they love water, and the tails are hydrophobic, they hate water, and the phospholipids are turned in on each other, so the outsides can appear in water and the insides are free of water. All right, as we move down, the first type of transport is called passive transport, and it does not require ATP, but is still influenced by other factors such as temperature and surface area to volume ratio. Examples of passive movement include ion channels that allow ions to flow along their concentration gradient either into or out of a cell. It also allows for diffusion to take place. Diffusion is the process by which atoms or molecules flow along their concentration gradient. It does not always need a protein, but most of the time it involves a protein in that it involves an ion channel. Then we have facilitated diffusion, in which large molecules still flow along their concentration gradient either into or out of a cell, but it uses an alpha helix, and the examples here are glucose or amino acids. Last of all is osmosis, which we can loosely define as the diffusion of water, but we'll talk about it later. All right, here we've got a diagram of diffusion in which ions are flowing through an ion channel into the cell in this case, in its passive process. Here we've got facilitated diffusion. We still have glucose or amino acids flowing along the concentration gradient, but it uses a carrier protein, but in this case it's passive and requires no ATP. Then we've got active transport. The word active means it requires the use of ATP. All right, the first example is what we call active transport, is where the movement of ions is against the concentration gradient, and again it uses an alpha helix because the alpha helix links the outside to the inside. Then under active transport, we've got something called bulk flow, in which we've got large amounts of material moving. We have exocytosis, in which a large amount of material is moved out of the cell using vesicles from the Golgi apparatus. Then we have endocytosis, where a large amount of material is brought into the cell, and again it creates a vesicle that's taken off to a lysosome. There are two types of endocytosis. There is phagocytosis, which is cell eating, and there is pinocytosis, which is cell drinking. Okay, and here are some diagrams of exocytosis where the vesicle binds with the cell membrane to release its contents outside of the cell. And here we've got endocytosis, where a large amount of material is wrapped in a vesicle and taken. Okay, then we move up to here. Whenever we're talking about the movement of water or ions, we need to talk about solutions. A solution is a solute plus a solvent. The solute is dissolved in the solvent, and the solvent inside a cell is water. We use comparison words. So these words are used to compare either side of the cell membrane. There is hypertonic. Hyper means high solute and therefore low solvent. Hypertonic means low solute and therefore high amount of solvent. And again, the solvent in a cell is water. Isotonic means equal solute concentrations on either side of the membrane. And here we've got 3 diagrams. This one shows you that's hypertonic on the outside and hypertonic on the inside. This is isotonic, which means there's the same concentration on either side of the membrane. And then we've got hypertonic on the outside and hypotonic on the inside. All right, now we think about osmosis. The definition of osmosis is the net flow of water. Net means the overall. So the overall direction of water flow from a high concentration of water, which we know to be hypotonic, to a lower concentration of water, which we know to be hypertonic. So we always say osmosis flows from hypo to hyper. It flows along the osmotic gradient, which is the concentration gradient for water, through a semi-permeable membrane, which is the cell membrane. Semi-permeable means it lets through some things like water, but prevents other things like ions from moving through. And it's a passive process, which means it does not require ATP. Now, when we're talking about water movement in plant cells and animal cells, it depends on what cell is in what solution. So if we put a plant cell in an isotonic solution, the cell is said to be flaccid, all right, because the cell membrane is not pushed up against the cell wall, and therefore it's a little limp. If it's put into a hypotonic solution, water will flow into the cell and the cell becomes turgid. The cell membrane will be pressing up against the cell wall and the cell will appear to be very stiff. If we put a plant cell into a hypertonic solution, all of the water will leave the cell membrane will move away from the cell wall, and we call this plasmolysis. If we put an animal cell in an isotonic solution, we say this is normal. If we put it in a hypotonic solution, water will flow into the cell will swell, and if it bursts, then we say that the cell has undergone cytolysis. If we put an animal cell into a hypertonic solution, water will move out, it will shrivel, and we call that process crenation. As we said before, there are other things that affect the rate of movement into and out of cells, and the first one is temperature. The higher the temperature, the higher the kinetic energy of the molecules, and osmosis will occur at a faster rate. If we've got a larger surface area, there is more area for the movement of particles, and therefore the larger the surface area, the faster the rate of diffusion. The concentration gradient is super important. The concentration gradient is the slope between the high concentration and the low concentration. The steeper this slope, the faster the rate of diffusion. The size of the particles also matters. Smaller particles will move through the cell membrane faster. Okay, so we're zooming out. Thank you for listening, and goodbye.